Hello viewers, welcome to my channel Biology Notes. Today I will discuss about spontaneous mutation. According to the mode of origin, gene mutation may be two kinds: spontaneous mutations and induced mutations. Today our topic is spontaneous mutation. Spontaneous mutations are naturally occurring mutations. They occur suddenly in the nature and their origin is unknown. All types of point mutations occur spontaneously. Spontaneous mutations can occur during DNA replication as well as during other stages of cell growth and division. It also can result from the movement of transposable genetic elements. In humans, the spontaneous mutation rate for individual genes varies between 10 to the power minus 4 and 4 into 10 to the power minus 6 per gene per generation. For eukaryotes in general, the spontaneous mutation rate is 10 to the power minus 4 to 10 to the power minus 6 per gene per generation. And for bacteria and phages, the rate is 10 to the power minus 5 to 10 to the power minus 7 per gene per generation. Most spontaneous errors are corrected by cellular repair systems. Only some errors remain uncorrected as permanent changes. In Gene Mutation Part 1, I had already discussed about tautomeric shift, base pair substitution, frame shift, etc. These all are happened during DNA replication and are responsible for spontaneous gene mutation. So here I will not discuss again. This type of DNA replication error may be repairable by mismatch repair systems. I will now discuss how chemical changes cause spontaneous mutation. Depurination and deamination of particular bases are two common chemical events that produce spontaneous mutations. Here I have drawn a diagram to show depurination process. Depurination is one of the most prevalent forms of DNA damage in which the N-glycosidic bonds are cleaved to release the corresponding adenine or guanine from DNA. This chemical reaction is especially predominant under acidic conditions in acidic gastric juice and some acidic organelles such as lysosomes. Thermal fluctuations also cause depurination in DNA. When depurination occurs with DNA, it leads to the formation of apurinic site. Apurinic sites formed in genomic DNA often induce replication errors and thus become potential sources of spontaneous mutation. Studies estimate that as many as 5,000 purines are lost each day in a typical human cell. Now, I will discuss deamination of DNA. Deamination is the removal of an amino group from a base. For example, in this diagram, the deamination of cytosine produces uracil. Now, this uracil is not a normal base in DNA. A repair system replaces most of the uracils in DNA, thereby minimizing the mutational consequences of cytosine deamination. However, if the uracil is not replaced, an adenine will be incorporated into the new DNA strand opposite it during replication, eventually resulting in a GC to TA transition mutation. Here is another example. DNA of both bacteria and eukaryotes contains small amounts of the modified base 5-methyl cytosine in place of normal base cytosine. Deamination of 5-methyl cytosine produces thymine, thereby changing the guanine 5-methyl cytosine base pair to the mismatched base pair guanine and thymine. Now, there is a big problem. 5-methyl cytosine deamination mutations are less likely to be corrected by repair mechanisms. So, locations of 5-methyl cytosine in the genome often appear as mutational hotspots, that is, nucleotides where a higher than average frequency of mutation occurs. So this is all about spontaneous mutation. If you have any query, please comment. Share the video if you like. Subscribe the channel for more new videos. Stay with me. Keep watching. Thank you.